Okay, welcome everybody. This is the chapter on our lymphatic system and our body defenses. And what we want to talk about today is what body systems keep us disease free. And if we look really at the body as an overall system, what we find is that it's the lymphatic and immune systems that are at the core of our defense mechanisms, but also we'll discuss a little bit about what we call our innate immunity, which are our body's barriers to infection, which include our skin, our mucous membranes, our secretions, and also our inflammatory response. So let's start by a discussion of the lymphatic system. It consists of two semi-independent parts, the lymphatic vessels and the tissues and the organs. And its job is to transport escaped fluids back into the blood, tissue fluid that's between the cells, eventually needs to enter the lymphatic system so that it can be screened for the presence of disease-causing entities that are called pathogens. This plays an essential role in our body's defense and our resistance to disease. If we didn't constantly filter our body fluids and remove pathogens as they manage to penetrate our outer barriers, then we'd quickly be overrun with disease and get sick very quickly. So if we look at the lymphatic system overall, what we find is that between most every cell in the body, there is a fluid component, and that fluid component is called the extracellular fluid. And it contains a lot of important stuff. It contains electrolytes, it contains waste products, it contains hormones, a considerable amount of water, and also um, products that the cell makes that it extrudes into the environment. Cell products such as components of the extracellular matrix, things like collagen and elastin, and also signal molecules, things like hormones. All of that's in the environment surrounding the cell, but sometimes we have unwanted material surrounding the cell, and here we're dealing directly with the pathogen. So the lymph represents the excess tissue fluid that's carried by the lymphatic vessels. The properties of these vessels are that they move in a single direction towards the heart and away from the tissue fluid. There is no lymphatic pump per se, but the way the lymph is moved through these lymphatic capillaries is through the action of skeletal muscle on the vessels. As the muscle squeezes and relaxes, the lymph is propelled through a series of one-way valves that eventually allows it to rejoin the general circulation near where the venous return to the heart and the lymphatic system hook up, which are right near the right and left subclavian veins. Um, there's also a rhythmic contraction to the tunica media in the lymphatic vessels that helps to squeeze the lymph along as well. Remember in our discussion of the circulatory system that blood vessels have three tunics, a tunica intima, a media, and an externa. The lymphatic vessels have the same three tunics, and so they also have a middle layer that's made up of smooth muscle. And what you're looking at in this diagram is just an illustration of how it is that the tissue fluid that may contain pathogens enters the lymphatic system. It does so as a result of differences in pressure between the tissue fluid and the fluid that's in the capillaries. We'll see in the next inset that the walls of the lymphatic vessels are made up of overlapping plates that will open inward in response to pressure and allow this fluid to get into the capillaries, where it'll eventually move through this milking action through one-way valves, making it ultimately to lymph nodes, which are organs that are going to contain very special cells that are designed, number one, to engulf and display little bits of pathogens called antigens on their surface, and the cells that respond to those antigens, which are called the lymphocytes. Okay, and then what they do is they populate these organs, and when they encounter an antigen that they recognize, this will stimulate the lymphocytes to divide. They'll build a standing army, and they'll go out and they'll actually fight the invader. Eventually, this filtered lymphatic fluid will flow probably through a series of lymph nodes, and then once it exits the lymph nodes, it will rejoin the general circulation, the venous return, up near the right and left subclavian veins, which run right underneath the clavicle. Lymph capillaries have walls that overlap to form flap-like mini valves. The fluid leaks into the capillaries, which are anchored to connective tissue by tiny filaments. The higher pressure inside closes the mini valves once they fill with fluid, and the fluid is then forced along the vessel due to the action again of skeletal muscle contraction. Okay, so you can see how the capillary beds and the lymphatic capillaries are very closely associated with each other, as they are with the surrounding tissue and the tissue fluid. And then what you're looking at with this diagram is how the tissue fluid moves into the lymphatic capillaries, and from there eventually is going to be escorted to lymph nodes, from there to lymphatic vessels, from there to lymphatic trunks, and from the trunks ultimately back to the venous return near the subclavian veins. And this is just an inset here showing you these valve-like structures that line the lymphatic capillaries that open in response to pressure differential and allow the fluid to enter the lymphatic system where it will be filtered. Now, these lymphatic collecting vessels collect the lymph from the lymph capillaries, and they carry it to and away from the lymph nodes and return the fluid to the circulatory veins near the heart. The major ducts include the right lymphatic duct, which drains into the right subclavian vein, and the thoracic duct, which drains into the left subclavian vein. And if we look here, we can actually see where the lymphatic system rejoins the venous circulation. You can see here, those are your internal jugular veins. There is the right subclavian and the left subclavian vein. You can see where the thoracic duct enters into the left subclavian and where the right lymphatic duct enters into the right subclavian. But I want you to notice something about the lymphatic system in general while we're looking at it here, which is that you tend to find lymph nodes clustered in regions of the body that are most likely to encounter pathogens. And so we see quite a few of them in the neck near the cervical region, quite a few under the arms in the axillary region. They're all around the digestive system in the abdominal pelvic region, a lot in the inguinal region, quite a few near the elbow, quite a few behind the knee as well. All of these drain fluid from areas of the body that are near ports of entry. Okay, obviously you've got quite a few up here in the head. You've got the respiratory and digestive system that run through the thoracic and abdominal pelvic cavities, and those are ports of entry, so we've got a lot of lymphatic tissue stationed around there, and that's why we have all these lymph nodes in the abdominal, pelvic, and thoracic region as well. And then, of course, we have to station lymph nodes in the extremities. The extremities are very likely to encounter pathogens as a result of use. They may become punctured or lacerated or abraded, and that could represent a port of entry for a pathogen. And so we'd like to have the ability to filter potentially infected fluid from these regions as well. The cisterna chyli right here receives lymph drainage from the digestive organs, and eventually it will drain into the thoracic duct, which is going to empty into the left subclavian. The right lymphatic duct receives lymph from the right side of the head, the right side of the thoracic cavity, and the right arm. The thoracic duct receiving lymph from the cisterna chyli basically drains the lymph from everywhere else in the body. Okay, so the lion's share of the filtrate actually comes through the thoracic duct into the left subclavian vein. Now, harmful materials will enter the lymph vessels, and that's by design, and these include pathogens such as bacteria, viruses, cancer cell, and also cellular debris. A pathogen is anything that has the potential to cause disease, and pathogens can either be living or non-living. In this particular list, okay, if we were to check off who is who, the living pathogens here include the bacteria, the cancer cells. Also, we should list fungi, parasites, um, and the non-living pathogens include viruses and toxins. Okay, and also um, we should list as cellular debris a non-living component that needs to be cleared by the lymphatic system as well. Cells eventually die, and this is one of the ways that we remove these waste products. So we have to defend ourselves against this onslaught because these harmful components are in the environment around us all the time. They're in the air we breathe, they're in the water we drink, and in the food that we eat. Now, you can see here how the lymphatic system is set up. This is the figure we were just looking at, and I want you to notice where the lymph nodes are stationed. They're designed to filter lymph before it's returned to the blood. Defense cells live inside these lymph nodes, the macrophages, which engulf and destroy foreign substances as they enter, and the lymphocytes, which respond to these presented antigens, and then will divide, producing an army that will go out and actually battle the infection effectively by generating a chemical attack that only targets that particular pathogen and not just a generalized chemical onslaught. So the lymphocytes, remember, are we're going to talk about our agents of adaptive immunity. So let's take a look at a lymph node in close-up. You can see that most are kidney-shaped and less than an inch long. They have an outer cortex, which contains follicles, which are collections of lymphocytes, and then there's an inner medulla, which contains phagocytic macrophages. And so the idea here is that the fluid enters the lymph node through these one-way vessels, and then notice that there are more entrances into the lymph node than exits, and this allows the fluid to temporarily collect inside the node. It flows through these open areas that are called sinuses. Okay, you can see the sinuses indicated here by these sort of brown staining areas in the diagram. Essentially, the fluid comes in, flows through these open areas, and these open areas, of course, are populated with macrophages. And so if there's a pathogen that gets inside, the macrophage will eat it, will chop it into little bits, and will display it to the lymphocytes that primarily populate here these other portions in the cortex, which contain a large population of lymphocytes. These are what are called germinal centers, which are constantly generating new lymphocytes on a daily basis. And what will happen eventually is that a particular lymphocyte amongst your billion different lymphocytes that have different specificities, meaning they recognize different antigens, a small population of those lymphocytes will be chosen for their ability to interact with the presented antigen on the surface of the macrophage, and they will actually begin to divide and produce now the army that's going to go out and actually specifically fight the disease. The filtered lymph then exits through the efferent lymphatic vessels and eventually is going to be returned to the general circulation. And so you've got these lymph nodes all over the body, and you've probably noticed that sometimes when you have an infection, one of these nodes will swell up and become sore, and that's a phenomenon that we call lymphedema, and it indicates that the body is actively fighting infection. So this is why sometimes when you go to the doctor, he'll feel under your jaw or under your armpit as a method to try and ascertain whether or not you're battling an infection, and it'll also give him some clue as to what area might be harboring the infection. So if we look at the lymph node flow through these nodes, what we find is that it enters the convex side through the afferent lymphatic vessels. It flows through a number of sinuses, and inside the node, a sinus is just an open space inside an organ that can contain fluid or gas or a combination thereof. The lymph then exits through the efferent lymphatic vessels. There are fewer efferents than afferents, and the purpose is to provide time for the lymph node to actually process the fluid that's coming into it and filter the pathogens out before they get a chance to make us sick. Okay, so uh, slow flow in this case is good flow. Now, there's other lymphoid organs that we need to address in our discussion of our body's defenses, and these include the spleen, the thymus, the tonsils, and the Peyer's patches. And you can see in the diagram here the location of each. You've got a total of 5 tonsils up here near the pharynx, which is the common chamber that's shared by air, food, and water. You've got a thymus gland that sits in front of the heart and behind the sternum, which is the site of T cell maturation. And then you've got a spleen, which sits lateral to the stomach in the left upper quadrant and is responsible for the recycling of red blood cells, and also serves as a lymphatic organ in that it contains macrophages and lymphocytes that can respond to pathogens that flow in it via the splenic vein, and also it will monitor and filter tissue fluid that's in the area as well. And of course, in the intestines, we have Peyer's patches that sit in the submucosal layer, waiting for any pathogen that might breach the mucosa, which is the inner lining of the digestive system. In the event that this happens, there are macrophages and lymphocytes in these clusters of tissue that always act as a team. Remember that the macrophages do the digesting of the pathogen and the presenting of the antigens, and the lymphocytes respond to those presented antigens and then generate now our standing army. The appendix actually turns out to be a very relatively important lymphatic organ. There are quite a few lymphocytes and macrophages that populate the submucosa of the appendix, in addition to its role as a safe haven for the bacteria that normally populate our gut, primarily our large intestine, which help us digest some of our waste products as they enter the last part of the gastrointestinal tract. So other lymph organs that we want to be aware of. Basically, wherever you find a port of entry, you're going to find lymphatic tissue, because that is really the best place to station these on-site emergency med techs. You could think of the lymphocytes and the macrophages as your body's emergency medical team. So let's talk about the spleen. It's located on the left side of the abdomen, and it filters blood, destroys worn-out blood cells. Remember that red blood cells don't have a nucleus, and so they can only make about 2.5 trips through the circulatory system before mechanical stress produces so much damage that they can't hold themselves together anymore, and so they need to be recycled. And so some components of the red blood cells are held onto and reused, things like the iron and the globin portion of the hemoglobin, and other parts are discarded. Those include some of the components of the heme, which is the actual organic molecule that houses the iron atom, which binds to the oxygen in hemoglobin. So that's its job really as a recycler. It also acts as a blood reservoir in the adult and a blood-forming organ in the fetus. By the time that we're adults, however, its role as a blood-forming organ has been abrogated to the role of the red marrow as our primary hematopoietic tissue. And just to revisit, remember that our red bone marrow is primarily in our bony thorax and in our pelvic and pectoral girdle, and in the heads of the femur, and also in spongy bone. So those are constantly making new red blood cells as the old ones wear out. And they're also a major site of formation for the most of the white blood cells as well, in addition to the lymphoid organs. So let's talk a little bit now about the thymus. It's located in front of the heart and behind the sternum. It functions at peak levels during childhood, where it produces hormones that help T lymphocytes become immunocompetent, meaning capable of fighting diseases. So you could think of the thymus as T cell school. There's only one class of incoming immature T cells that move into the thymus, and there's only one class that graduates from the thymus. And once they move out, they populate the lymphatic tissue throughout the body, and that's when they assume their role in fighting disease. Up until that point, when they're still being educated, it's important that the developing immune system get a little bit of help. This is why the fetus actually gets some antibodies directly from the mother while it's developing in utero, and also through mother's milk as it nurses, as a way to sort of buy time for the immune system to mature to the point where it can actually fight disease effectively on its own. This is why a lot of studies that show that babies that were brought up on formula tend to have a sicker childhood than those who nurse naturally, because there are no antibodies in formula like there are in normal human breast milk. So now let's look at your tonsils. They are stationed at a strategic point, a port of entry, the mouth. These are small masses of lymphoid tissue around the pharynx. They trap and remove bacteria and other foreign material, and sometimes they get overwhelmed and they can inflame, and this is what we call tonsillitis. They become congested with bacteria, and in the old days, what that would necessitate is an operation called a tonsillectomy. The reasoning being that at that point, the tonsils, instead of defending us against disease, are now serving as a source of infection, and so it's incumbent to remove them before they become necrotic and the infection spreads from the tonsils outward. Today, they don't go to tonsillectomies quite as readily. They try and treat with antibiotics. Back when I was growing up, the tonsillectomy was generally the first procedure recommended in the event of inflamed tonsils. And not to worry if you've had your tonsils out, you've got more than enough lymphatic tissue to keep you disease-free, even if you've had them removed. Now, your Peyer's patches are found in the walls of the small intestine, and they resemble tonsils in their structure. They just differ in their location. They're found in the submucosa of the small intestine, and they capture and destroy bacteria before it gets a chance to get into the bloodstream. Remember that they are collections of macrophages and lymphocytes acting as a team to keep us disease-free. And then we've also got lymphatic tissue in the mucosa. This includes Peyer's patches, tonsils, and other small accumulations of lymphoid tissue. They act as a sentinel to protect the respiratory and the digestive tract. Okay, so they're sitting there just beneath the most superficial layer of the respiratory and the GI. In the event that a pathogen penetrates, they can intercept it, ingest it, display the pathogens to the lymphocytes, and then the lymphocytes will divide and produce that standing army that will then go out and battle the disease. So if we look here at a chart showing you our body's defense system, what we see are really three lines of defense. The body is constantly in contact with bacteria, fungi, and viruses, and it has two defense systems for foreign material. Our innate or nonspecific defenses, which are using the same method of defense regardless of the identity of the pathogen, and behind them are our adaptive defenses, which sculpt a chemical attack based on particular characteristics of that strain of pathogen. So this is a much more targeted method of keeping us disease-free. Immunity is a specific resistance to a disease, and what's nice about immunity is not only is it very tightly directed against the pathogen, but it also has a phenomenon known as memory. Basically, once we successfully knock back a particular pathogenic assault, we store cells that remember that attack in our body for the rest of our lives, and in the event that that same pathogen shows up a second time, um, we have the standing army ready to knock it out before it ever gets a chance to produce disease, because it simply overwhelms it in terms of sheer numbers. And so you can see here in this chart how these different lines of defense operate. Our first line of defense, our skin and mucous membranes and their secretions, um, stand guard against the onslaught of pathogens that surround us all the time. And then behind them are our phagocytic and natural killer cells. There's also antimicrobial proteins in a lot of these secretions. And then, of course, we've got the inflammatory response, which you're all familiar with if you've ever had a laceration or a puncture. The wound will redden, will swell up, will warm up, and will hurt. And the idea behind the inflammatory response is to bring blood to the area of the infection. The blood contains white blood cells, and the white blood cells are disease fighters. The pain allows us to be conscious of the fact that we've had a trauma or an injury of some type, so not to use the affected area and give it time to heal. Um, the heat is designed to promote the metabolic activity of the white blood cells and inhibit the metabolic activity of any living pathogens. And the redness is there because the blood is coming to the area, and with it comes fluid, and that's what produces the edema. And it's the edema pushing on nerve endings that produce the pain signal that produce the pain that we associate with inflammation. And the nice thing about each of these lines of defense is that they don't share a weak spot. They're all different types of systems, and so the likelihood that you're going to get a single pathogen that's going to be able to penetrate all three lines of defense is relatively remote. If they all use the same mechanism to defend us against disease, the likelihood that they would be penetrated would be much higher. So it's nice to have these three methods of keeping us disease free all operating at the same time. And one of the things that you see in individuals that are lacking one of these lines of defense is that they're more prone to infection and disease, which is not a surprise because one of these lines of defense acting on their own aren't going to be sufficient to keep every pathogen out of the body and keep us relatively healthy. Now, your innate defenses are mechanisms that protect against a variety of invaders, and they respond immediately to protect the body from foreign materials, while our adaptive defenses are specific against a particular type of invader, and they're aimed primarily at a particular characteristic that invader has called an antigen. Okay, the antigen will identify the incoming pathogen as alien to the body. The result being that the immune system will recognize it as something that doesn't belong, and then will draft an army of lymphocytes that will be equipped with components on their cell surface that respond only to that particular antigen called receptors. Your innate body defenses are mechanical barriers to pathogens, such as the surface coverings of your body, including your skin and mucous membranes, and there are also specialized cells throughout the body that provide a barrier as well, including things like your ependymal cells that line the ventricles of the brain, okay, and other areas of the body that basically lock out pathogens getting inside. There are also chemicals that are produced by the body that inhibit the pathogen's ability to access our deeper tissues. These are things like lysozyme, which is an an enzyme that we find in saliva and tears and sweat that cleaves certain components in bacterial cell walls and causes them to lyse and burst apart. There are also certain secreted antibodies that can fight pathogens at ports of entry, such as globulins that we see in saliva, just to name a few. So let's talk about our skin and mucous membranes, our first line of defense. This is a physical barrier to foreign materials, and it also generates protective secretions. The pH of the skin is acidic, and this inhibits bacterial growth. The sebum is toxic to bacteria, and vaginal secretions are extremely acidic and prevent the movement of bacteria through the urinary tract. Also, we should point out that urine itself is a sterile solution that flushes the urinary tract of pathogens that are trying to gain access. So the more urination, the cleaner you keep the tract. The stomach mucosa secretes hydrochloric acid and also produces powerful proteases that inhibit bacterial growth. The saliva and the lacrimal fluid contain lysozymes, which is an enzyme that helps destroy components of the bacterial cell wall, causing it to weaken and eventually lyse in the presence of an osmotic shock. And then mucus traps the microorganisms in digestive and respiratory pathways and basically keeps them from moving in other places throughout the body. The mucus is also eventually expelled. It either gets spit out as you expectorate, or it gets swallowed into the digestive system where the digestive juices will help destroy the pathogen. So again, mucus, an excellent way to keep the inside of our body relatively pathogen free. There are also cells that help us remain disease free that use the same type of chemical attack regardless of the identity of the pathogen. Natural killer cells, the inflammatory response, phagocytes, antimicrobial proteins, and fever are all part of this second line of defense. And we'll break down what each of these is in the upcoming slides. So let's start with a chart that sort of shows you how these nonspecific defenses keep us relatively healthy. So natural killer cells are a type of white blood cell that patrols the body, and they have the ability to lyse and kill cancer cells, causing them to either disintegrate or dissolve. They can also destroy virally infected cells by releasing a chemical called perforin that punches holes in the cell's membrane and nucleus, and this causes the inner components of the cell to leak out, and the cell dies. Basically, it just withers away. You can see here sort of a flow chart showing you how this operates. This is the inflammatory response, which is also very important in keeping us disease free. You have the injured tissue that releases certain chemical components into the surrounding tissue fluid. These are things like histamine and other chemicals that are chemical attractants for macrophages and lymphocytes, but they also have certain local effects, causing, for instance, the blood vessels to dilate to increase blood flow to the area. The capillaries sort of open up, and this is to allow white blood cells that flow by to move through the capillaries and into the tissue fluid and battle the pathogen. Neutrophils and monocytes are attracted to these chemicals. They gain access by squeezing through these leaky capillaries and into the area of the injury. This leads to increased blood flow to the area, which causes redness and an increase in temperature. The edema that results from the fluid leaking into the area produces pain and swelling, and the swelling really actually is what produces the pain. And what this leads to is that we bring nutrients and oxygen into the area. This provides an excellent environment to support the metabolism of the leukocytes, which are disease fighters. And it also helps remove damaged tissue and cells from the area as they are basically destroyed. What happens is that the white blood cells move into the area and they begin the job of um, temporarily repairing near the site of the injury, providing a temporary scaffold called a clot, which eventually becomes a barrier of fibrin, and then the white blood cells will do battle with the pathogen that's locked in behind this fibrin barrier before it gets a chance to spread in the body. Um, in addition to this, there is limitation of joint movement due to the pain. This gives the entire process a chance to establish itself, and eventually this all leads to healing. And so what we see here in this inset is a look, the close-up of inflammation near the site of a wound, how all these components act together. Okay, so this is a diagram showing you the early parts of the inflammatory response. The goal is to prevent the spread of the pathogen. We don't want it to get into the bloodstream where it could produce septicemia. And it helps also dispose of cellular debris and pathogens through phagocytosis, and it also sets the stage for tissue repair. So you can see here that white blood cells enter the blood from the bone marrow. They roll along the vessel wall until they reach a point near the injury. There are components that are in the vessel wall that cause them to stick. These vessels will open up near the site of the injury, allowing the white blood cells to move through a process called diapedesis, where they'll be attracted to chemicals that are produced by the damaged tissue. This is positive chemotaxis. This brings them then to the area where they can phagocytize bacteria and other pathogens and prevent their spread. So what I want you to listen to here is a short inset talking a little bit about the inflammatory response and how it occurs, and then I will rejoin you in a discussion of how macrophages operate. Okay, so take a listen.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Okay, now that we understand a little bit better how inflammation works, we can address one of the other important functions that the immune system carries out, which is the destruction of pathogens and the processing of their antigens by phagocytosis. Cells like neutrophils and macrophages do this naturally. There are also cells such as dendritic cells that have this capability. What they do is engulf foreign material into a vacuole, and then lysosomes, which are cellular organelles that contain powerful proteolytic enzymes, will fuse with this vacuole that contains the pathogen and chop it into tiny bits. And those bits will be basically combined with a special type of a protein that's found on the membranes of every cell in the body called a major histocompatibility protein. And that combination of the antigen plus the major histocompatibility protein will be used to alert the adaptive immunity, the lymphocytes, to the presence of this pathogen inside the body. So you have to remember that the lymphocytes would have a hard time identifying pathogens in the body without the macrophages, and the macrophages, while engaging in phagocytosis, is a good defense against infection. It alone wouldn't serve to keep us disease free. So it's this other hidden function, the antigen presentation, that really allows the immune system, specifically our adaptive immunity, to keep us free from disease. And so what you're looking at here is simply a list of the steps that phagocytosis undergoes. It is a form of active transport, so ATP is used in this process. And you can see that there is vesicle movement from one side of the cell to the other, with subsequent fusion with the lysosome in order to digest the component parts of the pathogen. Now, there are also other types of chemicals that serve to defend us against infection and disease, and these are classes of proteins that are called antimicrobials. Antimicrobial essentially means anything that will attack and kill a microbe, which is a generic term for a bacteria. They attack microorganisms. They can hinder their reproduction. Most important chemical defenses include the complement system, which is a cluster of proteins that can puncture the cell membrane of an infected cell or an invading pathogen and cause the cytoplasmic contents to leak out and the cell to die. And these proteins can also trigger a series of chemical reactions inside the cell that cause it to commit suicide. Interferon is an antiviral that is produced by cells that are infected with virus, and what it does is it interferes with the formation of viral replication intermediates. Basically, it ruins the virus's life cycle. The thing about interferon, though, is that it's reasonably short-acting. It only affects cells that are near the infected cell, near neighbors, and it's not secreted in large quantity. And for the longest time, it was a riddle as to why this was. It was once thought that interferon, because of its antiviral and also apparent anti-cancer property, would be an excellent medication to give to people to rid themselves of viral infestation or to reduce the incidence of cancer. But it turned out that when these proteins or when these compounds were given in high doses to patients, that it caused damage to the liver cells, the hepatocytes. They were basically hepatotoxic compounds, and they then had to be pulled off the market. Complement, as we touched on earlier, is a group of at least 20 plasma proteins that are activated when they encounter and attach to alien cells, either those that are infected or those that come from outside into the body in an attempt to produce disease. What happens when complement is activated is that it damages the foreign cell surfaces, and it can release vasodilators that promote chemotaxis and chemicals that promote phagocytosis, and this is a phenomenon that's called opsonization. And basically, it allows the macrophages to more easily find the infected cell or the pathogen and grab onto it and digest it so that it can be destroyed. And what you're looking at here in this diagram is what we call the membrane attack complex which is part of the complement cascade. And what happens when these proteins assemble is that they will form a pore in the membrane, and that causes the cytoplasmic contents to leak out and the cell to die. This can be triggered by antibodies binding to antigens on the membrane of the infected tissue or of the invading pathogen. It can also be triggered directly by the presence of unusual components that are found on the surfaces of microbes. Components, for instance, of the cell wall can also trigger this response, among others. Interferon is a series of proteins that are secreted by virus-infected cells that bind to healthy cell surfaces and interfere with the virus's ability to reproduce. And fever is an abnormally high body temperature. The hypothalamus essentially becomes reset by the presence of pyrogens, which are chemicals secreted by leukocytes that cause the hypothalamus to promote now an increase in the body's overall temperature. The high temperatures inhibit the release of iron and zinc from the liver and spleen, and these are needed by bacteria in order to reproduce. And the fever also increases the speed of tissue repair. Okay, so several things going on here. Also, the high temperatures are not the ideal condition for many microbes to reproduce, whereas our own immune cells and our own tissues have no problem reproducing under these conditions of elevated temperature. The immune response is the immune system's response to a threat, and immunology is the study of immunity. One of the major tools in the immune system is the antibody, which I like to think of as a magic bullet. An antibody has essentially a Y shape to it as the basic structural unit. I can draw an example here, okay? And in every antibody there are at least two antigen binding sites. The simplest antibodies have two light chains bound to two heavy chains, and then the heavy chains and the light chains are bound to each other by disulfide bonds. And then the antigen binding region, I'll put here in blue, okay, is the part that actually attaches to the alien molecule. And so what this does when it finds a pathogen is that it will bind to its surface. That will cause complement fixation and lysis. But another way that antibodies can fight disease is through what we call immunoprecipitation or agglutination, where multiple infected cells or multiple pathogens become cross-connected to each other, and the entire complex comes out of solution and becomes an easy target for a wandering macrophage. So there's three aspects of adaptive defense. It's antigen specific, meaning that it recognizes and acts against particular foreign substances. It's systemic, so it's not restricted to the initial infection site, and it has memory, meaning that it recognizes and mounts a stronger attack on previously encountered pathogens so that we don't get sick with the exact same strain of pathogen a second time. Types of immunity include humoral immunity, which is mediated by antibodies, the magic bullets that we were talking about earlier. The reason it's called humoral immunity, you might wonder where this word comes from. The term humoral immunity addresses the body's fluids, okay? So let's put up here fluid, okay? And essentially it was once believed that our overall health was governed by the balance of our body's humors, our fluids. And so the solution to every disease during that period of time was to bleed you. And we did this either by cutting you or by applying leeches in an attempt to make you well again. And obviously, this didn't always work out. Back in those days, the local doctor was also the local barber, and he would dry the tools of his trade on a white pole mounted outside his office door, and these would include bloody rags. Okay, and so here we get the origin of what we know as the barbershop pole. Okay, so there would be our typical medieval barbershop pole with the bloody rags drying out in the sun. Okay, so this term has stayed with us all the way until modern times. The humors, the body's fluids, serve as the medium through which antibodies act. They're able to flow through the body's plasma and through the body's tissue fluid until they attach to a molecule called an antigen, which is its binding target. And at that point, it can promote complement fixation and lysis, or it can promote opsonization, which attracts macrophages to destroy it, and it can also promote agglutination or immunoprecipitation. It's provided by antibodies that are present in our body fluids when we're talking about this humoral immunity, and these are produced by a special type of lymphocyte called the B lymphocyte, which matures in our bone marrow. Cellular immunity, or cell-mediated immunity, targets infected cells, cancer cells, and the cells that come from grafting. And this is, again, one of the body's other mechanisms to keep us disease-free. And it works, again, on antigen recognition. If you have the wrong ID tag, so to speak, on your cell surface, you're going to be a target for these types of lymphocytes. They're called cytotoxic T cells that will home in on this particular unusual antigen and then target you for destruction. Okay, antigens are essentially ID tags. They're collections of molecules that, for the purposes of the human body, identify tissues as belonging to you. Okay, and in the immune system, essentially the antigens that are on your own cells are tolerated. As a result, we don't mount an attack normally on our body's own tissues, with very few unusual exceptions that we'll talk about in a bit. So an antigen is defined as any substance that's capable of exciting the immune system and provoking an immune response, provided that these molecules are not present on our own cells. Examples of common antigens include foreign proteins, nucleic acids, large carbohydrates and lipids, as well as pollen grains, and the surfaces of microorganisms, which have unusual molecules that make up, in some cases, their cell wall or components of their cell membrane. Self-antigens are on human cells throughout the body, and they identify those cells as belonging in your body, and so the immune system will tolerate them. Okay, we don't normally attack our body's own surface proteins. Our cells in another person's body, however, could be attacked because the likelihood that you're going to randomly find an individual in the general population who's going to have the same antigen display as yourself is extremely remote. The one exception would be identical twins. Other than that, generally it's difficult to mix and match tissue between individuals because of this antigen incompatibility, and this is what leads to graft rejection. This is why generally when we perform a transplant, we have to check the antigens on the donor tissue and the recipient tissue to make sure that there's a close enough match that the immune system will, at least to some extent, tolerate the transplant. This is generally found among living related donors, and that generally only happens when an individual needs to receive a paired organ, okay, or a portion of an unpaired organ that wouldn't kill the donor. Other than that, you're waiting for the accidental death of somebody who happens to be what's called histocompatible to you, and the likelihood of that happening on a day-to-day basis is extremely remote. So a lot of these people are on recipient lists for a long, long time. This restricts donors for transplants, with, of course, the exception, as we pointed out, of identical twins. Now, allergies are a special type of immune disease which we call hypersensitivity, where the immune system essentially overreacts to the presence of an unusual antigen inside the body. Many small molecules that are called haptens or incomplete antigens may not be antigenic, but when they connect with our own proteins, they can produce an immune response. What happens when the protein and the hapten couple up is that the immune system can recognize this as potentially a harmful entity. Okay, might be, as far as the immune system is concerned, attached to a pathogen. And so what can happen is that we can produce now antibodies that will recognize and destroy this protein hapten combination. And if this protein hapten combination is on the surface of our own cells, what this will do is it'll promote damage of our own tissues. Cells of the adaptive defense system are our third line of defense, and these include our B and T lymphocytes, which are designed to respond only to particular antigens from one B or T lymphocyte to the next. Macrophages, remember, are there to help the lymphocytes out. So one of the things that we have to understand about the immune system, this is a very important point to draw home, is the fact that when the immune system is being formed, what happens is that we have in the DNA of the lymphocyte population variations that allow it to produce a wide variety of antibodies and a wide variety of what we call T cell receptors, if we're talking about the T cell population. So many so that you've got close to 1 billion different possible antibody or T cell receptors that are going to be produced by a billion, up to 1 billion different types of B or T lymphocyte clones. So within everyone's body, we have an army of B and T lymphocytes, all that have a different mode of recognition, a different antigen that they're keyed in on because of the difference in the antibody or the T cell receptor that they produce. And it's the job of the immune response, the adaptive immunity, to find amongst those billion different B and T lymphocytes those particular lymphocytes that specifically recognize the antigen on the surface of the pathogen that's actually promoting the infection. And that may only be a tiny percentage of the total B lymphocytes. That subpopulation is then chemically stimulated to reproduce, and that's what's going to generate the army that's going to go out now and fight this particular infection. Now, if we want to sort of describe some of the characteristics of a lymphocyte, one of the things that we say about it is that it's immunocompetent. The cell becomes capable of responding to a specific antigen by binding to it. The cells of the adaptive defense system include lymphocytes, which come from hematocytoblasts in the red bone marrow, B lymphocytes, which become immunocompetent in the bone marrow, and the way to remember this is B for bone marrow, B for B lymphocyte, and remember that the B lymphocytes are the antibody producers, okay, the agents of humoral immunity, and then the T lymphocytes, which are chemically educated to fight disease in the thymus, that lymphatic organ that sits behind the sternum and in front of your heart. And what these guys do are several things. T lymphocytes come in three broad categories. You've got the helper T lymphocytes, which chemically stimulate the B lymphocytes, promote their cell division, and allow them to produce now massive amounts of antibody to fight the infection. They also, the helper Ts, stimulate the reproduction of the cytotoxic T cells, whose job it is to show up at the site of an infected cell or near an actual pathogen and to produce now a chemical attack that will compromise either the membrane surrounding the infected cell or pathogen, or actually promote the phagocytosis of the pathogen by macrophages, or produce chemical changes inside the cell that cause it to commit suicide. Any of these are possible methods that the cytotoxic T uses in order to do its job. And then the final class of T lymphocytes are the regulatory T cells, and what they do is they actually turn off the immune response once the pathogen has been conquered, so that we don't spend valuable resources, energy and raw materials, producing white blood cells to fight a battle that's already been won. We can go ahead and flip the off switch at that point and not worry about continuously stimulating these cells to divide and go out and engage in battle with a pathogen that's, for all intents and purposes, been rendered inert. Now, what you're looking at here is just a chart indicating where the immune cells go after they mature in the bone marrow. So we're talking here primarily about the genesis of what we call hemocytoblasts, which are capable of producing myeloid and lymphoid stem cell lines. The lymphoid lines will grow up to become the lymphocytes, and the myeloid line will produce the rest of the white blood cells in addition to the erythrocytes and the platelets. So if we follow the movement of these hemocytoblasts out of the bone marrow, lymphocytes that are destined to become T cells are going to migrate in the blood to the thymus and develop into immunocompetent T cells inside this organ. Um, the B cells are going to develop immunocompetency within the bone marrow itself, so they will not migrate to this organ that sits in front of the heart and behind the sternum in order to become capable of fighting disease. So remember, B cells maturing in the bone marrow, T cells maturing in the thymus. Once they become immunocompetent, they're still somewhat naive. They have yet to encounter antigens. They'll leave the thymus in the bone marrow and they'll start to seed the other lymphatic organs, which include the lymph nodes, the spleen, the liver, the Peyer's patches, and so on. In addition to the lymphocytes, remember that there are also going to be macrophages, antigen-presenting cells inside each of these organs, because they have to work as a team in order to effectively fight disease. Antigen-activated immunocompetent lymphocytes and memory cells are going to continuously circulate in the blood and the lymph throughout the rest of the body for the rest of the individual's life, depending on whether or not there is a need for them. Essentially, if they encounter a site of infection or site of injury, this will start the production of factors released from damaged or infected tissue that will tend to attract lymphocytes and macrophages to the area of the trauma in order to effectively stem the tide of the infection. Now, macrophages come from monocytes, which are a type of white blood cell that falls into a class known as an agranulocyte. They're called agranulocytes because they don't have visibly staining colored bodies inside their cytoplasm that are called granules. They tend to appear in a blood smear as a round cell with a purple staining kidney-shaped nucleus. They become widely distributed in lymphoid organs, and they're capable of secreting cytokines, which are vital in coordinating the immune response. And many of them, when they get to lymphoid organs, a fair number tend to remain what we call fixed, so that they don't leave those particular organs. They simply stand guard for fluid that passes through them that potentially can contain pathogens, so that they can engulf those pathogens, chop them into little bits, and then display those molecules, those antigens on their surface in combination with the major histocompatibility proteins. And so what you're looking at here is how the B cells help us fight infection. B lymphocytes with specific receptors are going to bind to a particular antigen in combination with its major histocompatibility protein. The binding event will activate the lymphocyte to undergo clonal selection, and a large number of clones will be produced, and this is what we call our primary humoral response. Most B cells will become plasma cells. These are going to be the major antibody producers in the plasma. They're going to generate the magic bullets called antibodies that will bind to the antigen and mark it for destruction, either through complement fixation and lysis, or through the triggering of opsonization, or through immunoprecipitation or agglutination. Their activity lasts for between 4 and 5 days, but during the course of fighting this infection, a portion of these B cells will be set aside as what we call memory cells. And in the event that they encounter this exact same antigen again, they'll be stimulated to divide and produce an overwhelming attack on the pathogen the next time it manages to penetrate our innate defenses and wipe it out before it gets a chance to make us sick. And this is what we call memory. So what you're looking at here essentially is the primary response here, where we have our initial encounter with the antigen on an antigen-presenting cell, such as a helper T cell or perhaps a macrophage. What happens as a result of this interaction is that the B cell is stimulated to proliferate. It'll begin to divide rapidly. That results in the production of these plasma cells and also, in the meantime, these memory B cells. And of course, once the infection is knocked back, what will happen is that regulatory T cells will help turn off this proliferative response, and only the memory B cells will remain for the next time that exact same pathogen manages to get inside the body and attempt to make us sick. And so what you're seeing here is an example of the primary and secondary humoral responses. Because the memory cells can live for decades and decades, a second exposure generates a rapid response, and this secondary exposure is going to be stronger and longer lasting. Note that the time elapsed here is in weeks, and what we're looking at here is the relative antibody titer in the plasma. And with the initial exposure, it takes 2 weeks to reach peak antibody production, followed by a long shoulder and decline. But with the second exposure, what happens is that before even a week is up, inside just a few days, we generate massive amounts of antibody in the secondary response, followed by a higher circulating level of antibody throughout the rest of the battle against the pathogen as we try to knock this infection down. And the result basically is that we don't get sick the second time around with the same exact strain of pathogen. Active immunity occurs when B cells encounter antigens and produce antibodies. And the reason that this is such an effective form of immunity is that these B cells are going to stay with us our entire life in the form of memory B cells, and of course during the active infection in the form of plasma cells. Active immunity can be naturally acquired as a result of an accidental exposure during the course of your activities of daily living, or it can be artificially acquired as a result of being vaccinated with an antigen that's purified from that pathogen, which will not produce the disease, but will produce an immune response that's capable of knocking the actual pathogen down should it attempt to infect. And so this is one of the reasons why vaccinations are so critically important in maintaining our health, because what they do is they prepare the immune system for the presence of the pathogen before the pathogen ever arrives, and so the pathogen never really gets a chance to produce the disease in an effectively vaccinated individual. Now, occasionally, from time to time, the immune system can age and atrophy a bit, and occasionally we have to have another vaccination in an attempt to remind the immune system that this antigen is present on a virulent pathogen, and we need to keep a standing army ready in the event that a subsequent infection shows up. An example of that is the recommendation of shingles vaccine for individuals that are approaching their golden years in their 60s, 70s, 80s, and so on. And again, the vaccine will not contain the pathogen. The vaccine will only contain a portion of the pathogen that's incapable of producing the disease. If you've heard that people contract a disease as a result of getting vaccinated for it, what's actually happening in that case is that you're seeing the results of what we call a hypersensitivity event that causes now an adverse reaction to a component that's in the vaccine that's part of the antigen cocktail that we inject, and that is a completely different phenomenon from the disease itself. Um, Different individuals all have different immune systems, and so we all potentially could generate a hypersensitivity reaction to any number of antigens that are out there in the environment, including antigens that you might find in a vaccine. So, in any vaccine for any type of disease, whether it be smallpox or herpes zoster, you're going to get a percentage of people in the population that are going to have what's called an adverse event due in part to this hypersensitivity response. So keep that in mind. Now, passive immunity occurs when antibodies are obtained from someone else. This can be conferred naturally from a mother to her fetus, where we call this naturally acquired. We get this, for instance, in breast milk, and we also can get it to some extent through the placenta while the baby's developing in the mother's uterus. And it can also be conferred artificially from immune serum or gamma globulin. In this case, we call it artificially acquired. Immunological memory does not occur in this case because what's happening here in passive immunity is that we're using borrowed antibodies to provide temporary protection from the disease until you're out of danger. Okay, you might, for instance, get a dose of passive immunity through a needle. Say if you were going to a part of the world that had malaria as an endemic disease, what you might want to do is get a shot of gamma globulin that's specifically directed against antigens that are found on the malaria parasite in order to provide you temporary immunity against the disease while you're in country. And then as the antibody is processed out, as it's naturally turned over, eventually what will happen is you'll be susceptible again. Okay, so this is what we call a prophylactic immunity. It's very temporary, and it only lasts while the antibodies are in your system. Now, monoclonal antibodies are prepared for clinical testing or diagnostic services, and they're produced from descendants of a single cell line. Uses for monoclonal antibodies include diagnosis of pregnancy and the treatment after exposure to hepatitis and rabies. And the beauty of a monoclonal antibody is that you get a cell culture that's capable of producing massive amounts of one particular antibody, one particular magic bullet that's very easy to purify and use for its specific purpose. These clinical applications are one example. We also use monoclonal antibodies as a way to detect the presence of antigens in histological samples. For instance, if we were looking for the presence of a particular membrane protein and we had no other way to find it, we might attempt to tag it with a monoclonal antibody attached to a dye in order to visualize it under a microscope using a special type of light source. They're also used to detect the presence of particular pathogens in a tissue sample or in a fluid sample or in a soil sample because they're directed against one particular antigen that is like a fingerprint for the presence of that particular microbe. And so here you can see a chart that sort of shows you how acquired immunity can be picked up. We can get it either naturally or artificially. If we're getting it artificially, we are getting basically the introduction of a component of immune resistance through a needle, okay, or through perhaps an oral in some cases, but generally it's an injectable. Artificially acquired active immunity results from being vaccinated using dead or attenuated pathogens, or these days, quite honestly, simply fragments of the pathogen that have antigenicity. And in passive artificially acquired immunity is the injection of gamma globulin, which contains antibody specifically directed against a particular antigen. And this only lasts for the amount of time that the antibody stays in your body fluids. Eventually, it's turned over and you become now susceptible again. Naturally acquired active immunity is where you have an exposure as a result of your activities of daily living, and you, as a result, generate a lymphocyte army capable of producing antibodies and T cells that are going to effectively knock the pathogen down. And the beauty of this is that you'll generate memory cells during the course of fighting this infection that will stay with you essentially for the rest of your life. Passively naturally acquired immunity is where we pick up antibodies from another source as a result of a natural act of development. And the two major events include receiving antibodies via the placenta during development from the mother's blood, or receiving antibodies from the mother's milk as you nurse. Okay, so two different ways to pick up now this passive resistance. Here you can see an example of an antibody. This is a soluble protein. This is one of the simpler looking antibodies. This is an IgE pictured here. These are secreted by plasma cells carried in the blood plasma, and they're capable of binding specifically to an antigen and promoting either complement fixation and lysis, opsonization, or immunoprecipitation and agglutination. And if you're wondering what part of the antibody is it that binds to the actual antigen, okay, it's the part at the ends of the Y-shaped molecule. So this would be an antigen binding region here, and this would be an antigen binding region here. And you can see very easily that in a typical antibody, it's capable now of binding to more than one pathogen at a time, and that's the mechanism that we know of as agglutination or immunoprecipitation. And what you're looking at here is just a diagram of antibody structure. You can see that we have two heavy chains with a variable region that's part of the antigen binding site, bound to two light chains that have an antigen binding site that sits directly opposite the heavy chain antigen binding site, and that both of these chains have what are called a constant region, which is the same from antibody to antibody and is not involved in the antigen binding process. They're held together by disulfide bonds, so this protein has both primary, secondary, tertiary, and quaternary structure. And remember that every antibody is going to be a little bit different with regard to this antigen binding site. There will be a different amino acid sequence that causes this site to have a slightly different shape, and as a result allows it to recognize different antigens. And you have approximately 1 billion of these, billion different antibody producing cells in a healthy functioning immune system. So there are also different classes of antibodies. Each class has a slightly different role. There are 5 major immunoglobulin classes. IgMs can fix complement. IgAs are found mostly in mucus, and IgDs are important in the activation of B cells, while IgGs can cross the placental barrier and fix complement, and IgEs are primarily involved in the allergic response. And the difference in each of these different classes are in the constant regions of the antibody and how those constant regions are attached to each other. They still all bind antigen. It's just a matter of where they bind antigen that's different from antibody to antibody in terms of location in the body where they encounter the pathogen. But other than that, they all essentially have the same structure that we saw in the previous slide, this Y-shaped structure with this constant region that serves either to anchor it to something, whether it be a cell membrane or another antibody, and this variable region, which is the part that actually binds to the antigen. Now, what you're looking at here is how it is that antibodies help us fight disease. They combat antigens in a number of ways. Complement fixation, where they initiate the complement cascade that eventually leads to cell lysis. Neutralization, where they essentially cover up a toxin so that it's not effective as a poison. And then agglutination, which we've discussed. You can see an example of agglutination here. And precipitation, which you can see here. I'm sorry, this is precipitation, this is agglutination, but the idea is the same. with agglutination and precipitation, where you produce such a large complex of antigen and antibodies that it's easy pickings for a wandering macrophage to come by and destroy it, knock it out, wipe it out. Okay, the cell-mediated immune response relies on the helper T cell in order to be properly chemically stimulated to divide and go out now and battle the the pathogen, as does the B cell. So what you're looking at here is how the whole process works. It begins when an antigen-presenting cell engulfs a pathogen and then chops the antigen into little bits and presents it on the surface in combination with a major histocompatibility protein. It's this complex that the T cell receptor will bind to if we have the proper T cell. Again, we have a billion different types of T cells in the body, each with different specificities. What will happen as a result is that there will be a chemical signal released from the antigen presenting cell that will stimulate the helper T cell to become activated. So what happens here when we recognize these antigens is that we recognize not only the presented antigen, but we also recognize the self proteins at the same time. This results in the stimulation of the helper T cell. It will release cytokines, which are soluble components that will stimulate not only the division of the helper T cell, but also the proliferation of the appropriate cytotoxic T and B cells that have the same ability to recognize the same antigen. And they do that as a result of interacting closely chemically with the helper T cell as a result of its ability to present the antigen to the cytotoxic T and B, and as a result properly stimulated. Okay, so there's a cell-to-cell touching that goes on in each case, first between macrophage and helper T, and then between helper T and cytotoxic T, or helper T and B cell. So what happens after antigen binding is that clones will form in the B and T cell population, and we'll go out now and destroy anything that has this antigen on its surface. So T cell clones that fight battle are known as cytotoxic T cells. They specialize in killing infected cells by inserting a toxic chemical called a perforin into the cell membrane. The helper T cells recruit other cells to fight the invader. These interact directly with the B cells. Regulatory T cells release chemicals that suppress the activity of T and B cells, and this stops the immune response in order to prevent uncontrolled activity, basically the wasting of resources fighting a battle that's already been won. A few members of each clone are going to become memory cells, so you're going to generate memory helper Ts, memory Bs, and memory cytotoxic Ts, all as a way to now produce a standing army that will be capable of knocking this pathogen out should it ever show up again. It won't get a chance really to make you sick. And what you're looking at here is just a chart showing you how the helper T cell plays a vitally important role in immunity by stimulating both the B cell and the helper T to proliferate, and then the helper or the cytotoxic Ts and B cells, which are capable of specifically interacting with the appropriately stimulated helper T, will then proliferate. The cytotoxic Ts, of course, will show up at the sites of infected cells or actual pathogens and generate a chemical attack designed to either compromise membrane structure, enhance phagocytosis, or promote cellular suicide, while the B cell's main modus operandi is to produce massive amounts of antibody that are capable of stimulating immunoprecipitation, agglutination, complement fixation and lysis, or opsonization, all of which can end up destroying either the infected cell or the pathogen proper. And of course, throughout this entire process, we're constantly generating memory B and T cells to stand guard should that exact same pathogen ever show up again. Now let's talk a little bit about organ transplants and rejection. There are several major types of grafts that we can use to replace damaged tissue in an effort to try and heal an area subjected to trauma or infection or both. Autografts are tissue transplanted from one site to another in the same person, while isografts are tissue grafts between identical individuals, and this can only happen in identical twins, basically two individuals that came from the exact same fertilized egg. And the reason for this is that they are antigenically identical, in addition to sharing a lot of other physiological traits between these two individuals. Allografts are tissue that are taken from an unrelated individual, and the only way that an allograft will work is if that unrelated individual is antigenically similar enough to the recipient that it's unlikely that a rejection will take place. And this is extremely rare. Generally, immunosuppressive drugs and anti-inflammatories have to be used in order to get the graft to take. Xenografts are tissue that are taken from a different animal species. Remember that autografts and isografts are ideal donors. Xenografts are rarely successful. Allografts are more successful with a closer tissue match. And again, it's very difficult for xenografts and allografts to work compared to autografts or isografts. This is why we generally have to use drugs that suppress the immune response in order to ensure success in the transplant. Now, hypersensitivity is essentially an abnormal, overly vigorous immune response to an antigen, which in this case is called an allergen. There's different types of allergies. Immediate hypersensitivity is triggered by a release of histamine from IgE antibodies binding to mast cells, which then degranulate and produce a massive histamine burst in local tissues. Reactions begin within seconds of contact with the allergen, and it can lead, in extreme cases, to a condition called anaphylactic shock, which is a dangerous systemic response to the allergen that can result in a massive drop in blood pressure, resulting in possibly coma and death. Delayed hypersensitivity is triggered by the release of lymphokines from an activated helper T cell. The symptoms usually appear 1 to 3 days after contact with the antigen. And what follows here are a series of videos that show you the different types of hypersensitivities and exactly how they work. So stay tuned and watch this little quick set of video clips. Okay, immunodeficiencies are a different type of disorder. This is where production or function of the immune cells or complement is abnormal. It can either be congenitally produced or it can be acquired. The major form of acquired immune deficiency is through infection with the AIDS virus, which attacks the helper T cell and basically abrogates its function, turns it into a virus factory that also has some of the properties of a suicide pill, in that every B cell and T cell that it interacts with ends up being destroyed. It causes it to commit suicide. So it's a double whammy when you pick up the AIDS virus and it affects the T cell population adversely. And essentially, when you lose your T cell function, infections begin to set up as they pass through our innate immunity that normally the immune system would pick off and destroy. But in the case of somebody who's immunocompromised in this way, these infections will begin to spread, and they're called opportunistic, taking advantage now of a weakened state in the immune system. The immune system, remember, does not distinguish between self and non-self. The body produces antibodies and sensitized T lymphocytes that attack its own tissues in autoimmune disorders. Examples of autoimmune diseases include multiple sclerosis, where the white matter of the brain and the spinal cord are destroyed, myasthenia gravis, which impairs communication between nerves and skeletal muscle, and type 1 diabetes, which destroys pancreatic beta cells that produce insulin. All of these result in the destruction of healthy tissue. And so the great riddle becomes with autoimmunity, why does this happen? How is it that the immune system makes this terrible mistake and starts to generate an antibody or a T cell clone that begins to recognize and destroy healthy tissue? And the answer is probably multifaceted. One way that this can happen is a mutation in the lymphocytes themselves, where it causes a change in the amino acid sequence of either the T cell receptor or a particular type of antibody that can, in some cases, recognize one of our body's own antigens. Another way that this can happen is through exposure to a cross-reacting antigen. You can imagine, for instance, that you might be out in the environment and you would encounter an antigen that is, let's say, 75 to 85% similar to one of your own antigens, but it does promote now the production of B and T cell clones that will attack and destroy it. And the problem with this is that once we've knocked back that particular antigen, we have lots and lots of B and T cells that kind of have nothing to do. And if they encounter your body's own antigen that is similar to this antigen that you just encountered and destroyed, it can bind to it, although not as effectively as its actual antigen, and initiate an immune response on healthy tissue. So this is why often we see autoimmune disorders cropping up in individuals who have experienced a recent infection, say with a virus or a bacteria, or a recent exposure to something in the environment. And then we start to see these autoimmune diseases often occurring in clusters. Other examples of autoimmune diseases include rheumatoid arthritis, which destroys joint tissue, and systemic lupus, which affects the kidneys, the heart, the lungs, and the skin by producing now these antigen antibody complexes that lodge inside blood vessels and impede circulation. And so you see a lot of very unusual symptoms with this disease. The disease gets its name, lupus, from a rash, a butterfly-shaped rash that spreads across the face, under the eyes, and above the mouth, that makes you look a little bit like a wolf. Okay, so lupus means wolf-like. Glomerulonephritis results in impairment of renal function. And again, this is an immune attack on the nephrons, the filtration units of the kidneys. We can also have a self-tolerance breakdown where we have insufficient lymphocyte programming, the appearance of self-proteins in the circulation that have not been exposed to the immune system can also promote an immune response. We see this, for instance, in eggs and sperm, in the lens of the eye, and in the cornea. And we also see this taking place in proteins that are in the thyroid gland, suddenly becoming exposed where before they were effectively isolated. Cross-reaction of antibodies produced against foreign antigens with self-antigens is probably the most common mechanism for autoimmunity. Examples of this include rheumatic fever, type 1 diabetes, lupus, and so on. So how is it that the immune system develops? Except for the thymus and the spleen, lymphoid organs are poorly developed prior to birth. A newborn has no functioning lymphocytes at birth, but only passive immunity that it gets from the mother. If lymphatics are removed or lost, severe edema will result, but the vessels will grow back in time. One of the most important things we have to understand about the immune system as it develops is that it's this passive immunity that the developing fetus gets from its mother in the womb through antibodies that are passed through the placenta. That's part of what buys time for the immune system to develop both in utero and in the neonate. After the baby's born, the source of antibodies comes primarily from mother's milk. And again, this is to give time for the lymphatic organs, the lymph nodes, the thymus, the spleen, and to some extent the liver, a chance to begin to produce immunocompetent B and T cells, and at that point the need for the antibody that is received by the infant passively through the mother's milk is no longer necessary, because now we have an immune system capable of fighting disease. This is why a lot of studies have been done that show that children who have been nursed on formula often have a more disease-ridden childhood than those that are brought up through natural breastfeeding. It's because of, in large part, this passage of immune globulin from the mother to the neonate that allows the immune system to develop, that allows them to have a healthier early several months of life. Okay, make sure to review this material, as it will be covered on the upcoming practical and lecture test, and I'll see everybody in class. Thank you.